The enzyme hexokinase is a transferase, isomerase, oxidoreductase and a ligase, which is the correct option. Before we select the correct option, let me just give you a brief highlight as to what are the different classes and what are the commonly used examples under these classes. So, class number 1 is called as the oxidoreductases. So, the name is suggesting you oxidation and reduction reactions. Any of the enzymes which take place parts in the oxidation and the reduction reactions, we would call them as oxidoreductases. So, when you see the word dehydrogenase, now here I have given you the example of lactate dehydrogenase. So, we know there's pyruvate dehydrogenase, there's glyceraldehyde 3-phosphate dehydrogenase. When we see the word dehydrogenase, we know they take part in oxidation and reduction. So, be safe and say it is class number 1. Now, even when you see an enzyme with oxygenases, oxidases, even they belong to class number 1. Now, coming to class number 2, we have the transferases. Now, what is transferases? Now, if you have an enzyme, for example, I have given you the example of glucose. Now, glucose gets converted to glucose 6-phosphate. The enzyme might be glucokinase or it could also be hexokinase. From where has this phosphate added to the glucose? So, the phosphate will be transferred from the ATP. So, ATP is acting as a donor of that phosphate. So, this phosphate will add to the glucose and give us glucose 6-phosphate. So, what has happened meanwhile? The phosphate has been transferred to glucose to give us the product glucose 6-phosphate. Because it's taking part in the transferring reaction, we would call them as the transferases. Now, any other examples do we have for it? Yes, commonly we even have the transaminases like the ALT and AST also belong to this class of enzymes. Coming to the third enzymes, that is the hydrolases. Now, hydrolases are the enzyme, the word is hydro, so they add water and then break a bond. So, when a enzyme works by addition of water and then breaks a bond, we call such enzymes as hydrolases. So, our digestive enzymes mainly belong to this particular class of enzymes, that is class 3. So, here I have given you the example of lipases, a digestive enzyme for the lipids. Next, coming to the fourth group, that is the lyases. Now, lyases are the group of enzymes which will break the bond but without the addition of water. That's the difference between class 3 and class 4. Class 3 uses water, class 4 breaks a bond but does not use water. So, common example I have given you here is fumarase. Now, class number 5, isomerases. Isomerization reaction. So, if you have an enzyme which reads like a isomerase, or even if you have racemase or if you have epimerase, all this belongs to class number 5. Class number 6 is ligases. When you have an addition of ATP and then you join two compounds, we call it as a ligase. So, ligase is to bind or to ligate, which is derived by an energy molecule, that is ATP. So, the example for this is DNA ligase. Now, kinases also belong to this particular group. Now, if you ask me why not hexokinase, does it become a ligase? No, because hexokinase, what is happening? The group is transferred to the glucose or a hexose and then you get a hexose 6-phosphate or a glucose 6-phosphate. Here what is happening is two groups are added. Example, if I have to give you, I can give you acetyl-CoA carboxylase. Now, what does this enzyme catalyze? It will catalyze the conversion of acetyl-CoA to malonyl-CoA. Now, this requires ATP and biotin and carbon dioxide. So, ATP is needed. It's an energy molecule that is needed here. So, there is no transfer of the group over here. So, such classes of enzymes which use ATPs are called as the ligases. So, if you have the enzymes which reads as synthetases, not synthase, synthetases, they also belong to class 6. So, we commonly use the mnemonic for this OTHIL, OTLIL. O stands for oxidoreductase, T stands for transferase, H stands for hydrolase, L stands for lyases, I stands for isomerase, last is ligase to bind. Okay, that's the 6th class. Now, translocases is a new addition which is the 7th class of the enzyme. Translocase is different from transferase. Translocase is change of a location. Okay, transfer of a location. So, where we have commonly ATP synthase or we have ADP, ATP translocase. Now, when we need a translocase is when we have a different membrane. So, from one membrane to another membrane, like for example, a mitochondrial membrane, from the outer to the inner, or from cytoplasm into the mitochondria. If you need a translocation of the ions, such class of enzymes are called as the translocase. So, we have another T added to this particular classification of enzymes. Alright, now let's have a look at our question back again. The enzyme hexokinase is a transferase, an isomerase, oxidoreductase, ligase. I told you hexokinase. Whenever you see something like this, try to get where or which is the reaction it is catalyzing. So, hexokinase is a transferase. This is the correct answer. Not an isomerase, not a oxidoreductase because commonly it's a dehydrogenase, not a ligase. Ligase would have a suffix generally of a synthetase or a ligase. Alright.
To watch the full video, download our app on App Store and Play Store.